Start show. Okay. Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. In this video lecture, we are going to talk about fluoroquinolone. Fine. In the previous video lecture, in part first of the fluoroquinolone, if you remember, we talked about in a detail means the mechanism of action of fluoroquinolone. We talked about the drugs which are related to fluoroquinolone, the structure of the fluoroquinolone, DNA replication, and we also talked about the bacteria which are susceptible to the fluoroquinolone. Now this is the second part of the fluoroquinolone, and in this second part of video of the fluoroquinolone, we are going to talk about the mechanism of resistance of the fluoroquinolone. Fine. What are the mechanism of resistance of the fluoroquinolone? And we also talked about the side effect of the fluoroquinolone as well. Fine. So let me write. Mechanism of resistance. Mechanism of resistance of fluoroquinolones. Fine. Mechanism of resistance of fluoroquinolone. Now, if bacteria want to survive, so definitely bacteria will be survive. Fine. Now in environment, bacteria want to survive. Fine. And what actually they do? Then they will create some resistance. For example, if we are the bacteria, so definitely if we want to survive in the environment, so we should have to resist against antibiotic or against some other things which are harm to us. Fine. Similarly, bacteria will also create some resistance for their survival against these drugs. So the first resistance which are activated against these drugs, these are the mutation, and usually we call that spontaneous mutation. In spontaneous mutation, what actually happen? Some nucleotides in genes, some nucleotides will be changed. Fine. So let me write the first one, resistance. First one is spontaneous mutation. Fine. So first one is spontaneous mutation. Let's suppose in this case what actually happened. Let's suppose these are the genes. Fine. These are the genes of the bacteria, and let's suppose that area, let's suppose that area, there are some few nucleotides at that area. These few nucleotides means changed at that area. So what actually happened, we call that spontaneous mutation. Fine. And after that, an enzyme is formed. We get a resultant enzyme, and this enzyme is also topoisomerase enzyme. I'm talking about the topoisomerase enzyme. So then another topoisomerase is formed, but this topoisomerase will be altered. We call that altered topoisomerase, or we call that altered gyrase. So this is the structure. In previous video lecture, if you remember, the structure is totally different. Fine. So how the structure will be look like? If you remember, I draw some like something like that, or maybe different. But this is the structure which is not mutated. This is a topoisomerase which is not mutated. But the drugs means this is, let's suppose, the drugs. This is the fluoroquinolone. Fine. So in this case, this fluoroquinolone is attached here. They have the affinity. But what actually happen in this case means if the mutation occur, but in this case the drugs lose their affinity. They come, but they not bind. They not bind here. They lose their affinity. This is the first resistance which are activated against this fluoroquinolone. Fine. If you remember, this one area is responsible for the joining, because these are the two subunit. If you remember, these are the two subunit. One we call that gyrA, and other we call that gyrB. Fine. These two are the subunit. Fine. This one subunit is responsible for the joining, and this one subunit is responsible for the cleavage. Fine, if you remember. And I also told you that cleavage function is performed by endonuclease enzyme, whenever the DNA is replicated, and the ligation action is performed, or the ligation mechanism of action is performed by DNA ligase enzyme. But in this case, topoisomerase will perform both functions. They perform joining as well as cleavage, fine, to release the tension in a DNA. So this is the first thing. Now, so this is the first resistance which are activated against this fluoroquinolone. Fine. And then after that, what actually happened again, let's suppose this is the DNA. And these are the super coiling, let's suppose. Fine. Again, if they show resistance, so again they will sit here. They will sit here. And what actually they do? They will cleave both strand and join the strand as well to release the tension. Fine, I already discussed it in the previous video lecture. But now they will again do their job, and again they do, and after that what actually happen, if this is the bacteria, let me draw, if this is the bacterial cell, fine, and in this case if these are the circular DNA, because bacteria is usually contain circular DNA, fine. So this is the DNA, and after that this DNA will be replicated and make another copy of the DNA. Then after that what actually happen, if if the DNA strand, or we can say if the DNA are formed, replication is occur, then what actually happen, then from this one cell, from this one bacterial cell, two cells are formed, and each cell carry the same copy of the DNA, means this one DNA. One will present the old and the other will present the new. Fine. So this case again occur when the drugs are not perform their function, when the drugs lose their mechanism of action. So all these process will again happen, and then infection is again started in the within the body, within the human body. Fine. Now these resistance always occur. The resistance always occur in bacteria when we giving the antibiotic at the same time for a longer period of time. So antibiotic, we should have to give antibiotic on that time when the patient need. But when the patient doesn't need antibiotic, then we don't give the antibiotic. Fine, because when we give the antibiotic for longer period of time, then the bacteria will show resistance. Otherwise, bacteria will not show the resistance. So this is a very important thing you should have to keep. Antibiotic, we should have to give antibiotic for shorter period of time, for low amount, due to which the bacteria will die. But when we give it for longer period, so it will create side effect also. I will discuss it for later. Fine. Now this is the first resistance. Let me tell you second resistance. If you remember efflux pumps, I already discussed it in beta-lactam antibiotic. If you watch that video, if you go to my channel and watch that video, I already explained it in a very detail. What actually happened, once again let me tell you, second resistance is a flux pump. A flux pump production. In this case, what actually happened? Let me again explain for your understanding. Let's suppose this is a bacterial cell. Now, this red color, let's suppose this one represent the porine channel, and this area represents the efflux pumps. Efflux pumps will not present always, right? It will be present in case of resistance. So after that, what actually happened? Let's suppose this blue one represents the drugs, means the fluoroquinolone. This fluoroquinolone will get internalized, fine. But what actually happened, due to the presence of this efflux pump, they will receive very fast, very forcefully, with a very high force, they receive this antibiotic and expel out it from the cell. And then drugs will again expel out from the cell. So just think, when the drugs are not get internalized, although it internalized, but they again expelled out. So how the function will be performed by these drugs? So it is very important thing, fine. So again, the drugs will not perform their function, and again the bacteria will survive, and this topoisomerase will come here and they will do their job, and everything in bacteria will be fine, and then bacteria will survive and divided and infection is occur. So this is the second one resistance. This one resistance is always present in every case, whenever in every antibiotic cases, for example fluoroquinolone, beta-lactam antibiotic, and some other antibiotics, this efflux pumps is very important. Means this efflux pumps is against these drugs, against the antibiotics. These efflux pumps are very important, fine. So in usual or in some kind of bacteria or in many bacteria, these efflux pumps are very important for these antibiotic drugs, fine. So that's all about the. Now let me tell me some other thing about the fluoroquinolone. When we are giving these drugs at the same time again and again, then there are some side effects of these drugs. So let me write. These side effects are the insomnia, dizziness, headache, fine. So that's all about the fluoroquinolone. I hope this video helpful. If you like this video, please hit the like button, share this video, and also subscribe to my channel to get more interesting video lecture like that. Thank you so much.